Hello and welcome to this showcase session, which gives the opportunity for our audience to engage with users of nanopore sequencing, today specifically on neurodegenerative research, neurodegenerative disease research. Each of our speakers will give a brief overview of their work and their research interest, and then I will join them in a discussion. We will also be inviting questions. This is an online-only session, so we will only ask questions from the floor. So I'm delighted to welcome our first speaker, Joanne Trinh. Joanne heads the Integrative Omics in Parkinson disease research group at the Institute of Neurogenetics in Lübeck in Germany. The group investigates the role of mosaic variant, nuclear and mitochondrial genome sequence, and lifestyle and environmental factor in Parkinsonism. Thank you, Joanne.
Thank you very much, Cora. And thank you so much, Nanopore, for inviting me for this panel discussion. I'm very honored to be here today, very excited to discuss neurodegeneration in the context of long reads. And today I'm actually representing the monogenic network of the Global Parkinson's Genetics Program, or GP2, in their long read sequencing initiative. I myself am from the University of Liverpool, Institute of Neurogenetics. And our research group looks at integrative omics approaches in Parkinson's disease specifically. We have been looking at genetic and environmental modifiers in hereditary Parkinsonism, with a focus on mitochondrial genetics, repeat expansion, structural variants, looking at gene-environment interactions, and we heavily established the long-read sequencing platforms at the Institute to investigate these aims. A few highlights that we have, and I just wanted to touch base on that, are mitochondrial somatic variants that distinguish between monogenic forms of Parkinson's disease, like PINK1 and Parkin. We have discovered mosaic divergent repeat interruptions. And in addition to this, there has been a follow-up of the mosaic repeat interruptions by Joshua Lass, who is a PhD candidate in the group. And he has been wonderful, and he actually has a poster here today, poster number 6, if you are all interested. And in addition to this, we're looking at mitochondrial polygenic risk scores and how that is related to the environment. In terms of the Global Parkinson's Genetics Program, it's a big resource program by ASAP, the ASAP initiative. It's improving our understanding of the genetic architecture of Parkinson's disease worldwide and making this knowledge really globally relevant. And GP2 really aims to collect samples, 150,000 individuals now all around the world with Parkinson's disease. And we're planning to genome sequence these individuals across diverse populations. And this effort is led by Andrew Singleton and Cornelis Blauendrath. Within GP2, it is separated into a complex network and a monogenic network. And I myself am within the monogenic network, also co-led by Christine Klein and Nicolo Mencaci. Here are some highlights of what has been coming out of GP2. One is the identification of genetic variants and loci that are in admixed populations of African descent, and also multi-ancestry GWAS in Parkinson's disease. More recently, there's also a RAB32, a monogenic form of PD, a new gene that has been found for PD in collaboration with GP2. I just wanted to touch base on how long-read sequencing is related to this. We are very interested in looking at families with an unknown genetic cause of disease and trying to figure out what the genetic cause is and discover new genes. We are planning to do whole genome sequencing of 10,000 individuals, families or cases with early onset PD, as well as long-read sequencing on 1,000 individuals that are unsolved. And we're hoping that this will achieve rapid identification of new causal genes for PD. Thank you very much, and happy to take questions.
Thank you very much, Joanne. So I'd like to now welcome our second speaker, who's Kimberley Billingsley. Kim leads the long-read team at the NIH Center for Alzheimer and Related Dementia in the US. Her work focuses on generating large-scale, high-value dataset to study the impact of structural variant and tandem repeat in neurodegenerative disease.
Thank you, Cora. Thanks for a great introduction, and a big thanks to Nanopore for inviting me here today. And it's a delight to talk about our project where we've got up to production scale at CARD, and we're now doing long-read sequencing of about 100 samples a month. So the diseases that we work on, so these neurodegenerative diseases, even after decades of really large-scale genetic studies, we still don't know, as a whole, a lot of the genetic variation that contributes to disease risk. And one of the main reasons for this is that we can't, with the older technologies such as short-read sequencing and SNP arrays, we can't access much of the genetic variation in the genome, which we know that we can better access with longer reads. But the problem when we started this project a couple of years ago is there wasn't any scalable methods for doing long-read sequencing of these large disease cohorts. So the focus of my team's work has been developing these methods so that we can do scalable sequencing and to get these methods out in a way that they can be accessed and used by the wider research community. So we now have developed protocols where we can do high-quality nanopore sequencing from blood, brain, and cell line tissue. All of those protocols are on protocols.io. And then with these protocols, we can then develop these and generate these neurodegenerative disease data sets for everybody to use. And so along with developing the wet lab protocols, we've also developed a pipeline in collaboration with Benedict Payton at UCSC and Misha Kolmogorov at NCI. And so this is called NAPU. And the idea is to try and standardize the way that we're all analyzing nanopore data so that we can compare across data sets. And so with this pipeline, we have variant calls, we also have haplotype resolved methylation calls, and we also have de novo assemblies. And then we can use these new data sets and new variants to then do analyses like QTL analyses to look at what's the functional impact of these new variants that we discover. And so here's just a really brief overview of the data sets and the results that we've generated so far. And so I oversee two large-scale initiatives. So the first is the CARD Brain Initiative, and this is where we're sequencing thousands of human brains from Alzheimer's disease and related dementia cohorts. And so we've completed our first two cohorts. The first is the NABEC, which is European ancestry, and the second is the HBCC, which is African ancestry samples. And so far in this analysis, we've been able to identify thousands of new structural variants that were invisible with the short-read methods. And we've also been able to do QTL analyses where we've looked at the correlation between these genotypes and RNA-seq expression in the brain. And we've already been able to identify variants that are driving gene expression in the brain at known Alzheimer's disease and related loci. And the second project, which ties in with the project that was just discussed with the Monogenic Hub, is the work that we do as part of GP2, which is the Global Parkinson's Genetics Program. So we've just finished completely sequencing our first large-scale Parkinson's cohort, which is about 750 samples from cases and controls, and this is from blood. And so we were in the process of base calling this, but with the release yesterday, I think we're going to have to go back and rebase call those. And so in the meantime, what's been really cool about this data is we've also been able, in the meantime, identify patients that had Parkinson's disease, but we've actually found pathogenic expansions in those samples that are causative of ataxia. And so these large-scale initiatives are mainly focused on sporadic disease, but we've also done a lot of work in the monogenic space. And so after this talk, Guillem's going to give a great talk on the work that he did at CARD as part of looking at monogenic cases with these methods. Thank you.
Thank you very much, Kimberley. So we can start taking questions from the floor. We have a mic who's going behind you.
Thank you very much for those great talks. I guess I have a question for you, Joanne. You displayed, RAP32 paper that you recently published. I think this is a quite good example to see how, with GP2, we can find new genes or new variants, in this case of Parkinson's disease. So can you tell us a little bit more about this case, please?
Yeah, I can definitely tell you. So actually, RAP32 was found 7 years ago, which is a very long time ago, and we've continuously worked on it for 7 years. How it was identified was through an exome sequencing screening of Tunisian families, and a lot of consanguineous families we had in Tunisia. And then we found RAP32 as one of the hits. And then another Tunisian family had the same variant, this S71R variant. And then in addition to this, when looking at Canadian families, we found yet another family. So we had 3 families at the time in 2016, and we thought this was a really convincing evidence already that segregating, this variant segregates with disease, and it's right at the phospho site of this RAB32 with, so it has some functional consequence to this. And then we thought, okay, so how to follow up on this? But unfortunately, it did take a long time to look at this functionally. And in order for us to really find that link between LRRK2 kinase activity and this RAB32S71R, it took some time. But this is a very striking, very interesting link between LRRK2 and RAB32. So they interact. And what this mutation does is it affects the kinase activity, this autophosphorylation site of LRRK2. And as we know, LRRK2 G2019S, which also has this impact on the autophosphorylation site, it is already implicated in PD, and it's the most common mutation in familial PD across the world. And just to talk more about GP2 and how it's related, so once we have found these 3 families, then looking into other databases and spreading the word around, we found another 14 additional carriers, and one of which is within GP2. So that was really exciting that GP2 can really help in these sorts of gene lookup efforts. Yeah.
Another question from the audience? Go ahead.
It was for Kimberley. I was just wondering about the NIH CARD consortium. Have you called DNA methylation or any cytosine modifications on that data?
Yeah, so we're looking at methylation. So the first analyses that we've done is kind of individual CPG sites, promoter regions that have smoothed out the methylation and genes. And then we're doing like a QTL, but like an MQTL analysis with the kind of structural variants. Because of the scale of the project, I think we still need to work on tools to do like allele-specific and differential methylation in these really large population data sets. But at the minute, kind of the MQTL is the first analyses that we're doing.
Right. And will that data be made publicly available at some point?
Yes, we're working on the manuscript at the minute, and we're hoping that the data release will be pushed in June.
Right. Thank you.
And you're looking at 5MC and 5HMC, or?
Just 5MC.
Just
now,
5MC
yeah.
at the moment. Any other question? 1 minute. One more question. If we don't have one from the floor, I can ask one, because you're both looking at very similar disorder, but in very different cohorts. One is way more complex, later onset. One is much more earlier onset, familial. What do you think there is the specific nanopore strengths for those type of cohorts?
Yeah, so for the forms that I'm looking at, so we're trying to find the causal gene in these monogenic, potentially monogenic families. And these families have been looked at through exome sequencing, genome sequencing, and we've looked at it all throughout, but we haven't looked at the structural variants with long reads. So we think this is something that is really missing and could be that link that could explain all of these unknown cases. For example, synuclein duplication and triplication, these are pathogenic variants for PD. There are some Parkin duplications, deletions, but ever since exome came out, we've been really looking at single nucleotide variants and focused on that. So I think that there's much more to be found.
And you, Kimberley?
Yeah, so for the sporadic data set that we just generated for about 750 samples, so this is really high quality in terms of the N50s are around 35 kb and the coverage is about 40x, so it's going to be a really powerful data set. Just this nanopore was probably the only scalable option to do that. It took us 2 years of processing the DNA, kind of doing the sequencing, and hopefully those nice long N50s now will give us a chance to do de novo assembly and pull variants that we can't even identify with all the long-read methods.
Cool. We are out of time. So thank you very much, both. You can reach out to Kimberley and Joanne during the conference. And thank you again, both, for a very fascinating discussion.